So today's lecture is a continuation of our discussion of the molecular mechanisms involved in genetics. This slide is one that we've seen previously, and I just want to recap really briefly what's going on or what's depicted on this particular slide. Across the top, what you see is what's referred to as the flow of information with regard to genetics. We again have discussed in previous lectures that DNA is in fact the storage molecule that contains the genetic information. Essentially, this information is instructions to synthesize proteins that the cell needs to function. Now, that's a very simplistic view of what DNA is. There are other components that are not necessarily just instructions to synthesize proteins, but we'll get back to that in just a little bit. In our last lecture, what we focused on was the replication process. Replication is essential for allowing for cell division to take place. In both mitosis and meiosis, all of the genetic material has to be copied or duplicated or replicated in order to allow for the passage of genetic material to the new cells that are being produced as a result of cellular division. But today's lecture is a continuation of this flow of genetic information in that the DNA, like I've mentioned in a very simplistic view, is instructions to synthesize proteins that the cell needs to survive or function. What happens in the next step of the flow of genetic information is that genes are copied on a gene-by-gene basis in a mechanism known as transcription. Transcription is a process by which RNA molecules are synthesized on a DNA template. This process of transcription results in an RNA molecule that is a copy of a given gene. Now, transcription, like I just mentioned, occurs on a gene-by-gene basis, and so it is a very different type of copying mechanism than DNA replication that I just mentioned previously. DNA replication is a copying mechanism where the entire genome or genetic information is copied in preparation for nuclear or cell division. This lecture will focus specifically on transcription and the molecular mechanisms involved in transcription, but I also want to summarize the continued flow that you see on this particular slide. Once a gene is transcribed or copied, where an RNA copy is in fact made, in most cases, the RNA is utilized to synthesize a protein. It is that RNA copy that is instructions to synthesize or put together a specific protein. That protein then results in some final functional phenotype that we can assess overall. Now, not every single gene is what we would consider a protein coding gene. There are some genes that do in fact get transcribed and produce functional RNA molecules, and we'll see some examples of that later on in this particular lecture. The second part of this particular lecture will focus on the mechanism known as protein synthesis or translation. In translation, you have the RNA copy of a gene that is utilized as the instructions to synthesize a protein. So the definition of translation is the formation of a protein directed by an RNA molecule. So to continue summarizing the flow of genetic information, I like to refer to this particular figure. If you focus on panel A, what you see is a small portion of a gene that is encoded in the DNA. Here you can see the specific nucleotides that make up this small portion of a gene. What happens during the flow of genetic information is this gene is copied in the mechanism known as transcription. Transcription produces an RNA molecule, and you can see the sequence of that RNA molecule in yellow in the center of the slide. Now, with transcription, what happens, in summary, is that one of the strands of DNA is utilized as the template to synthesize the RNA molecule. What you can see very clearly in this figure is the sequence complementarity that holds true in this transcription mechanism. The RNA that is produced is typically referred to as mRNA or messenger RNA. That is from a subset of genes that are referred to as protein coding genes. There are some genes, like I mentioned in the previous slide, that are not protein coding genes. They do in fact code for functional RNA molecules. And again, we'll look at some examples of that in a few slides in this particular lecture. However, what we focus on mostly for transcription and then translation are genes that are referred to as protein coding genes. The transcript or mRNA that is produced is referred to as messenger RNA or mRNA. And for a protein coding gene, the copy that is made, the mRNA, is then utilized as instructions by ribosomes to synthesize a protein. In this flow of information, in the bottom portion of panel A, you can see the mechanism of translation. The nucleotide information that is found in the messenger RNA is specifically utilized as the instructions to synthesize a protein. 3 nucleotides at a time are referred to as a codon, and essentially these are like 3-letter words that are instructions to insert a specific amino acid. So in this example, you can see the codon UUA. That is the instructions to insert the amino acid leucine. The next codon, CAC, is the instructions to insert the amino acid histidine. And then lastly, the codon GGC is the instructions to insert the amino acid glycine. And this is a very simplified view of what happens during transcription and translation. In the center of the slide, panel B, you can see, referring back to some of our original genetic terms that we discussed in transmission genetics, in that you have a genotype that results in some phenotype. The genotype is dictated by what genes are present in an individual, and the phenotype is the expression of those genes. So let's look at panel C now. In panel C, you see the same flow of information, the gene in red undergoing transcription to produce the messenger RNA, and the messenger RNA then used during protein synthesis or translation to synthesize a protein. However, in panel C, what we're looking at is an example of what might be a different allele for a given gene. The sequence is slightly different compared to that in panel A. Specifically, the second nucleotide in panel C is a letter G. In panel A, it is an A. So there is a nucleotide difference in these two sequences, which again could represent different alleles that are present in a given individual or in a population. In this very simple example, that different nucleotide does in fact get transcribed, which results in a different nucleotide insertion in the mRNA that is produced. And the result in this specific example is that codon, the very first one, now is UCA. UCA codes for the insertion of the amino acid serine, which is different from what we saw in panel A. The codon was UUA, and it coded for the insertion of the amino acid leucine. So here we can see differences that occur in nucleotide sequence that can potentially result in differences in amino acid sequence that gets inserted into a protein. In this specific example, we have 2 distinct types of proteins that would be produced, which might be representative of different alleles. This table is referred to as the genetic dictionary or the genetic code. It contains all possible 3 nucleotide combinations that we refer to as codons. Every possible 3-nucleotide combination is listed here. I will never ask you to know these specifically, meaning you don't have to have each codon memorized for which amino acid it codes for. But what you should be able to do is read this information and determine if a codon was given to you, what does it in fact code for. There are some additional components that we will come back to when we get to protein synthesis, and that is the codon known as AUG, which is the start codon for protein synthesis, and 3 codons that are known as stop codons for protein synthesis, which are UAA, UAG, and UGA. But again, we will come back to that when we get to protein synthesis. So we're going to slowly transition into talking about transcription, which is that copying mechanism that occurs on a gene-by-gene basis. However, I always like to take a step back and review some basic concepts that hopefully are familiar to you, but are a little bit of a review. In this table, you can see a comparison of DNA and RNA. So when we talk about what DNA and RNA are composed of, as far as their building blocks, they are made up of nucleotides. DNA and RNA together are referred to as nucleic acids. The type of sugar in the nucleotides does differ between DNA and RNA, and I suggest you go back to looking at some of the figures from DNA structure where I highlighted this difference. DNA contains a sugar known as deoxyribose, and RNA contains a sugar known as ribose. So the nucleotides are slightly different with respect to the sugar that is present. There is also another difference in those sugars that is specific to the name, and that is in the number 2 position of the sugar, there is no hydroxyl group in DNA, and that hydroxyl group is present in RNA. The bases that we can find in the nucleotides that make up DNA are A, C, G, and T. And in RNA, it is A, G, C, and U. There are no T-containing nucleotides in RNA, and vice versa, there are no U-containing nucleotides in DNA. The linkage of nucleotides in DNA and RNA is essentially the same, nucleotide after nucleotide after nucleotide, and the bonds that link these nucleotides are referred to as phosphodiester bonds. Typically, DNA is found in a double-stranded form, which is the classic double-stranded helix that we think of when we discuss or think about DNA. RNA, however, is typically found in a single-stranded form. However, there are some complexities that we will eventually talk about for RNA. Now, the right hand of the slide really just focuses on that flow of information that we discussed in the first slide. DNA being copied in a transcription process to produce RNA, and RNA then utilized to synthesize a protein in most cases. What I did want to highlight from this are the types of RNA that can be produced by transcription. So the 3 largest classes of RNA that are produced by transcription are messenger RNA, which I've already mentioned. That is the product of transcription of a protein coding gene. The other 2 large categories of RNA that can be produced by transcription are those known as rRNA, which is short for ribosomal RNA, and tRNA, which is short for transfer RNA. Both rRNA and tRNA are utilized in protein synthesis, and we will come back to that in just a little bit. I also mentioned at the beginning of this lecture that most genes are considered protein coding genes, and that when they are transcribed, the transcript is referred to as messenger RNA, and the messenger RNA is utilized as instructions to synthesize proteins. Ribosomal RNA and tRNA are not protein coding genes. The RNA product from transcription is utilized in protein synthesis, but they themselves are not instructions to synthesize proteins. There are other genes that are considered non-protein coding genes that do undergo transcription and produce different classes of RNA that are functional RNAs inside the cell. And in the center on the left-hand side, you can see a list of some of these RNAs. Here too, I'm not going to ask you for specific names. These all have different functions inside of the cell, and that would be something that we might discuss in molecular genetics course if you were interested in taking that in the future. The last thing we need to discuss before we transition into transcription is what I refer to as the molecular description of a gene. Genes have components associated with them that are really important for allowing the genes to be transcribed. In this figure, you see a number of components. You see the double-stranded DNA, where the gene is located. In transcription, only one copy or one strand of DNA is copied, and that is referred to as the template strand. That is the one that is used as the instructions to synthesize the RNA molecule during transcription. What you can also see in the yellow block is what's referred to as a promoter. And a promoter is a specific set of sequences that allow for the initiation of transcription to begin. This is the site where the enzymes involved in transcription bind and begin the transcription process of this particular gene. At the opposite end, in the orangish color, right after the orange block, is what's referred to as the transcription termination site. So just like the promoter is a place to begin transcription, the termination site is where transcription will end. The result of transcription is the production of an RNA transcript. And what happens during transcription, just like with replication, is synthesis always occurs in the 5' to 3' direction. So the very first nucleotide synthesized in transcription is the one on the left side of the RNA transcript where the 5' end is. And the last nucleotide is at the opposite end on the right-hand side of the RNA transcript, also referred to as the 3' end. Just as we had done previously when we discussed replication, we focused on the mechanism found in atypical prokaryotic systems such as E. coli, and then added in a few additional informations about eukaryotes. We will continue doing that with transcription. So what I want to start with then is looking specifically at the enzyme that's going to be involved in the main process of transcription in E. coli. You can see on this particular figure a cartoon version of the enzyme and then the ribbon model associated with the enzyme as well. The enzyme is actually made up of two distinct subunits, or two different components, I should say. There is, if you look at the cartoon version in green, the sigma factor, and then the rest of the enzyme is considered the core. In the core, there are multiple subunits, which we'll talk about in just a minute. With what you see on this diagram, the sigma factor and the core together is referred to as the holoenzyme or the complete enzyme. The sigma factor can dissociate from the core, and the core will continue on with the transcription process. So let me go ahead and focus on the sigma factor first. The sigma factor is necessary or required for the initiation of transcription. It is also referred to as a specificity factor, meaning that it does recognize specific sequences within the promoter, like we discussed in one of the previous slides, and it associates with that sequence to drive the rest of the polymerase to the target site and allow for transcription to begin. The core, like you see in red, is responsible for elongation. There are multiple subunits found in the core. There is beta, which is required for initiation and elongation. It essentially adds nucleotides during the transcription process. Beta prime, which functions in binding to the DNA template, so allowing for that core component to associate with the DNA. There are 2 copies of the alpha subunit, and the alpha subunit is what allows the binding between the polymerase and the promoter. Essentially, it holds everything together. And then what's not shown on this figure is the omega factor, or the omega subunit, and it is the omega subunit that acts as a helicase. We discussed the importance of a helicase previously in replication, and that the helicase job is to unwind the double-stranded DNA. X-ray crystallography of this particular enzyme has allowed us to get a little more detailed picture of the structure of the enzyme. If you look at the ribbon model, it looks very similar to the DNA polymerase we discussed previously in that it has a hand or claw-like appearance to it. Similarly, this enzyme, the RNA polymerase, grasps the DNA. You have a channel in the center where it's the catalytic component. This is where nucleotide uptake occurs. This is where the newly synthesized RNA exits, as well as the DNA entering in that is being transcribed. Now what we'll do is take a little closer look at the promoter, which is the specific set of sequences that allows for transcription initiation to occur of a given gene. In the previous slide, we looked at that overall molecular view of a gene, and we discussed briefly the concept of a promoter, but here we're going to look at it in a lot more detail. In order for transcription to initiate, in order to get that process all started, you have to have association of the polymerase with a specific location that indicates start transcription here. And that's essentially what the promoter does. It's a set of sequences involved in the control of the expression of a given gene, and specifically it is the site where the RNA polymerase binds. What we have in this figure is what I would consider a core promoter. It is a typical or characteristic promoter that can be found in association with multiple or many types of genes. Most promoters will have what is referred to as a 10 base pair sequence that is upstream of the transcription start site. So in this figure, there's a little arrow all the way to the right that indicates where transcription of this particular gene begins. To the left of that arrow is the promoter. So the transcription start site indicates the +1 nucleotide when transcription occurs. Anything to the left of that, before the actual transcription start site, is called upstream of the gene, and these numbers have negative numbers associated with them because we're moving away from the transcription start site rather than into the gene. So at the -10 base pair position, or 10 base pairs upstream of the transcription start site, is something that's referred to as the -10 box. There is also a set of sequences 35 base pairs upstream of the transcription start site, known as the -35 box. Both of these sets of components are part of the core promoter, and what was done by some previous studies is to compare thousands and thousands of promoter sequences, and when doing that, it was very easily deduced that there are some sequences that are important, that are, again, important for transcription to begin. This is referred to as a consensus sequence, and a consensus sequence really is just the probability that a given nucleotide is in a specific position in a set of sequences. So oftentimes The minus 35 box, the minus 10 box is referred to as the ta-ta box. And in this case, it's because it has a number of T's and A's in it. Now, again, it is not a specific sequence found in every single promoter, but there is what's referred to as a consensus found in every promoter. And there is a majority of T's and A's found in this particular location. Strong promoters have a very high percentage similarity or identity to the consensus. Weaker promoters deviate from that sequence a bit. In addition to the core, some genes have what are known as upstream elements or up elements as listed on this particular slide. Again, not every single promoter contains these upstream sequences. Some do, and these typically play a role or some type of regulatory role. Most upstream elements in prokaryotes stimulate transcription by helping with the binding of the RNA polymerase to the sequence. And we'll see some examples of that in a little bit. Now, what I also want to add is the importance of a typical core promoter. A typical core promoter has what I refer to as 3 basic functions. The first one is really just to act as an on-off switch to allow for transcription to be turned on or transcription to be turned off. Essentially, that minus 10 box and that minus 35 box are what the sigma factor recognizes and binds to, the sigma factor in the RNA polymerase. So by having a strong consensus sequence in the minus 10 and minus 35 box allows for a nice interaction between the sigma factor that's part of the RNA polymerase and this core promoter. So simply having the sequence there allows for the sigma factor to bind, and this then allows for transcription to be turned on. If there is not a strong set of sequences there, then transcription is not turned on because the RNA polymerase doesn't bind as efficiently. Associated with that is the concept of what I refer to as a speed switch, and this just really means how efficiently transcription is occurring. Simply turning on or off transcription is one thing, but the speed switch is really efficiency. How efficient is the polymerase at binding, and how quickly can it associate and start transcription? If there is a really nice, strong consensus sequence in the promoter that allows for a good, strong interaction between the sigma factor and the gene itself, this will allow for very efficient transcription so that the polymerase binds and starts transcribing. Then as soon as that polymerase moves out of the way, the next one can bind and start transcribing. So this is what I refer to as a speed switch. It really is efficiency. How much output are we getting with the polymerase associating and transcript being produced? Lastly, the promoter also serves to align the polymerase in the correct location to allow for transcription to always begin at the same site. This is what I mentioned previously as the +1 transcription start site, and this is where transcription begins every single time for this particular gene. What allows that to happen is the specific alignment of the minus 10 and the minus 35 sequences, and the polymerase with those sequences line up nicely so that transcription always begins at the exact same location every single time transcription begins. All right, let's go ahead and jump on into the transcription mechanism or process. In transcription, again, the promoter is a specific set of sequences that allows for transcription to initiate. Specifically, there are some additional factors known as transcription factors that help recognize this sequence. For our purposes, we're not going to focus too much on these transcription factors. What we will focus on is the main enzyme involved in this process, known as RNA polymerase. RNA polymerase synthesizes RNA in a 5' to 3' direction. It is enzyme that synthesizes RNA, and so in this process does not require a primer like DNA replication did. In addition, the RNA polymerase is a multi-subunit complex in that one component of the RNA polymerase acts as a helicase. So if you go back and recall the replication process, what had to happen is the two strands of DNA had to separate before any replication could take place. This is also true for transcription in that the two strands must separate before transcription can occur. So the helicase subunit of RNA polymerase does just that, pulls apart the two strands, allowing for the polymerase then to recognize the single-stranded template and begin synthesizing RNA. So let's put this all together. You have the promoter of the gene that is recognized by additional protein transcription factors as well as the RNA polymerase. Those components engage with the promoter to initiate transcription. The helicase component of the RNA polymerase unwinds the DNA and allows for the polymerase component to begin synthesizing the RNA transcript. The RNA polymerase will continue to move away from the transcription start site towards the terminus, and as it continues to move in the slide from left to right, you can see the RNA transcript being synthesized during the process as the polymerase is moving towards the right of the slide. Lastly, the polymerase will reach the transcription terminus, and with the help of some additional protein factors or enzymes, will terminate the process. Essentially, what happens is the polymerase falls off of the DNA. The transcript is now separate. The double-stranded DNA is back in its double-stranded form, and now the transcript can be utilized for whatever it needs to be utilized for inside the cell. This particular figure is a really important one because it brings back this concept of sequence complementarity that I said a few lectures ago would come back over and over and over again. The sequence complementarity we discussed with respect to the two strands of DNA refers to what we also call the base pairing rule, and that is C's will always hydrogen bond with G's or G's with C's, and A's with T's or T's with A. This sequence complementarity is what dictates what sequence of RNA is going to be synthesized during the transcription process. The only difference is that there are no T-containing nucleotides in RNA. They are substituted with U-containing nucleotides. So if there is an A in the DNA template, it will be a U that is inserted in the RNA that is being produced. So you can see highlighted in this figure the DNA template down at the very bottom in the orangish color, and the RNA that is being synthesized in the yellow in the center of the slide. The process again occurs in a 5' to 3' direction, just like the synthesis of DNA during replication, and it is dictated by sequence complementarity. So now that we've looked at how transcription occurs in a little bit more detail in the previous two slides, what I want to do is go back to initiation and talk about initiation in a little more detail. If you focus on the left-hand figure, we'll get to the right-hand figure in just a minute, but with the left-hand figure, what you're looking at is what's meant to represent the RNA polymerase and the DNA sequence that it is going to associate with and begin transcribing. What happens is the holoenzyme very loosely binds to the DNA, and that's because the sigma factor interacts with those core promoter sequences that we just talked about previously. The sigma factor locates the promoter, and once that association occurs, the helicase begins to unwind the DNA. Here you get a very short DNA sequence that is in an open complex formation, and you can see that in the figure sort of in the middle of the figure. You have a small portion of the DNA unwound, which then allows for the single strand to act as the template for the synthesis of the RNA in the transcription process. However, what happens in this situation is that polymerization, the addition of nucleotides, RNA nucleotides, one right after the other, occurs. However, it typically occurs in very short stretches. This is referred to as abortive initiation. And this just means that there needs to be a very stable complex formed in order for the polymerase to clear the promoter and actually enter into transcription. So what you see in this figure with those little green stretches in the figure represent very short stretches of RNA that were synthesized, but the polymerase didn't continue. This is the abortive phase. You need to have a very stable complex between the polymerase, the DNA template, and the growing chain. And essentially what you get is a chain that's just long enough for the polymerase to keep on going and continue with the polymerization process or elongation. Once that occurs, the sigma factor dissociates. This is referred to now as promoter clearance, and the core can continue with the transcription process, adding nucleotide after nucleotide throughout the elongation mechanism. So as you can imagine, this process of initiation actually is a little bit difficult in order for the polymerase to continue on and allow for elongation to occur. So how can this be helped to make this a more efficient process? Well, one way to do that is to have parts of the polymerase, and in this case, we're going to look at the right-hand figure right now. In the red or orangish color are the alpha subunits that I mentioned when we discussed the structure of the polymerase previously. The alpha subunits have a dumbbell shape to them, in that there is what's referred to as the CTD, which is the C-terminal domain, and the NTD, which is the amino terminal domain. These are just designations of where the amino terminus versus the carboxyl terminus is of the protein that's making up this subunit. That part's not really important. What is important to understand is that there's a portion that you can see, the NTD, which associates with the core polymerase, and the CTD, which is associating with an upstream element. What this does is allow for a nice tight interaction with the upstream element and the C-terminal domain of the sigma factor, and this essentially anchors down that polymerase to get a nice tight association to ensure that promoter clearance can in fact occur and transcription continues on into the elongation phase. So if you have a gene that has an upstream activating sequence or an upstream element, and you have a polymerase that has a sigma factor that can recognize that upstream element, this can make transcription much more efficient. And we will look at some more specific examples of that in our next lecture. So we've discussed the initiation of transcription. We've discussed very briefly the elongation, which is just the sequential addition of nucleotide after nucleotide following that DNA template and the synthesis of the RNA. What we need to discuss now is termination. How is it that this process is stopped at the end of what would be the gene terminus? So the termination of transcription, you have the polymerase reaching the terminus or terminator sequence. Essentially, the polymerase falls off of the gene and the RNA is released. But this is a little simplistic view of what's occurring. There are two different types of mechanisms that we'll look at in just a minute. One is referred to as being row independent, and the second one is known as being row dependent. Row is a factor that plays a role in certain types of transcription termination. So there's either row independent, does not need that factor, and there's row dependent, does need that rho factor. Regardless of which type of transcription termination we're talking about, both mechanisms utilize something known as an inverted repeat and a hairpin formation. So on this slide, we're going to talk about that in a little more detail. So an inverted repeat is just a set of sequences that is repeated one right after the other. And so if you look at the top figure where there's the sequence, if you look at the top strand, which is 5' at the left and 3' at the right, if you follow the sequence T, A, C, G, A, A, G, where the dot is, then T, T, C, G, T, A. This is the inverted repeat. What that means is that you have the set of sequences to the point where the dot is, and then you have the complement after that dot. How this becomes important is if you look at what happens when you transcribe that specific set of sequences. When you transcribe that specific set of sequences, there are regions that have sequence complementarity, like you see in the bottom portion of that figure, and the dots just indicate where hydrogen bonding can occur. So in an inverted repeat, you get the sequence transcribed. Once it is transcribed, because of the sequence complementarity, a hairpin structure can form. And a hairpin is kind of like a bobby pin. It just indicates that those sequences can come together. And these are really important in the termination of transcription. And we're going to look at those in just a minute here. So let's look at rho independent, which just means it's a mechanism that doesn't require the help of an additional factor named rho. There is an inverted sequence in the terminator, followed by a A-rich region in the sequence as well. So you have an inverted repeat, then you have an A-rich region. What happens is the polymerase continues to synthesize RNA, and when it gets to the stretch of the A-rich region, it actually slows down just a little bit. So let me take one step back. The inverted repeat sequence is transcribed, and then when the polymerase gets to the A-rich region, it slows down or pauses just a little bit because it's kind of got like a little bit of a hiccup in the reading of the template. That slowing down allows time for the hairpin to form, and that's what you're seeing in the center of this slide is the hairpin structure forming. When that hairpin structure forms, either one of two things happens. There's either a physical tug of the RNA out from under the polymerase, or the RNA is essentially pushed away. In this mechanism, the research that has been done suggests that it's more of a physical tug. So as the hairpin forms, because of the stretch of A's and U's that follows it is very weak, that hairpin forming essentially pulls the RNA out from under the polymerase. And then this then causes a complete dissociation of the complex. So essentially you're getting the RNA that's being synthesized pulled out from the polymerase then falls off, and transcription is terminated. Pretty straightforward mechanism. So a separate mechanism for termination is known as rho-dependent termination, and it requires a factor known as the rho factor. The rho factor is what you see on the right-hand side of the figure with the ribbon model. It has a ring shape associated with it, and it's made up of 6 subunits. One component of this rho factor is an ATPase, and just to recap, ATPases are enzymes that cleave ATP, releasing the energy associated with ATP to perform some activity. And there is also an RNA-DNA helicase, which is a helicase that specifically pulls apart RNA-DNA hybrids. So if you go back to the left-hand figure, you can see a more idealized version of what's happening in this mechanism. In the first figure, what you see is the Rho factor binding to the transcript, specifically at a set of sequences known as the Rho loading site. It's typically 60 to 100 nucleotides in length. It's rich in cytosines, but it is a set of sequences that the Rho factor recognizes. So this is in the RNA. The Rho factor recognizes it and jumps on or loads onto the RNA that's being synthesized. It begins to travel up the transcript, moving from the site that it joined up towards the polymerase. This is why that ATPase activity is so important, because there's going to be energy involved in allowing for that enzyme to travel from the initial location it bound up towards the elongating polymerase. Now the polymerase is still synthesizing, but what happens is the Rho factor sort of catches up. There is also an inverted repeat sequence that is synthesized and a hairpin forms, and you can see that in the center of the figure. And when that hairpin forms, that allows the Rho factor to catch up to the polymerase, and the thought is that the Rho factor knocks the polymerase off of the template and dissociates the whole complex. And this is how Rho-dependent termination occurs.